Right. So, just continuing the last discussion, what we have discussed in the last lecture. See, the last discussion was all about, we were talking about the cloning, and in this head cloning, we have discussed two different approaches of the cloning. One was the recombinant DNA cloning, or DNA cloning. Suraj, video is perfect. So I hope that you are okay. It's okay, yaar. Don't bother. Don't bother, right? So, right. So the last discussion was all about the cloning and what we have done so far. We have discussed about the cloning, what actually the cloning is, and in this context of the cloning, we have discussed about the recombinant DNA cloning or the gene cloning, and we have discussed this therapeutic cloning as a stem cell cloning. And this stem cell, we have discussed three different types. One is embryonic stem cell, second is adult stem cell, and this third is iPSC, that is the induced pluripotency stem cell, right? So we have done this one. Nikhil, in the last class, we were discussing about the plant genomics, not in the last class, it was the last to last class. It was on Friday class, right? In the Friday class, we have discussed about the plant genomics, right? And in the Saturday class, we discussed about the human genomics, and in the context of the human genomics, I shared PPTs to everyone where we started with the human genome project. We discussed about a number of genomic projects across the world, and in that context, we have also discussed about the different genomic projects which has already been carrying on in India as a national genomic grid, like Indigene project. Fine. And then after doing this entire discussion, we have discussed about the gene therapy, right? Then again we have discussed about the stem cell technology. So this much of discussion already we have done in last class, that was on the Saturday, right? And then this stem cell technology was the last discussion, and this part was the left part of the last discussion as a reproductive cloning, and this reproductive cloning is nothing but it's a organism cloning. So here the last discussion, I don't know who communicated you. Sunday class was, today's Monday, na? So Sunday class was cancelled. Means Sunday there was no class. I don't know who communicated you. And we have done this class on Saturday. Right? Saturday we have done class till the 7:05. So anyway, I don't know who communicated you. So this is the last discussion that we are going to do. We are going to start here. That is the reproductive cloning, right? And this reproductive cloning is also called as organism cloning. Reproductive cloning is also called as the organism cloning, right? So this involves cloning of an organism, and when we talk about the cloning of an organism, so this cloning could be in the case of the plant, this cloning could be in the case of animal, right? So the simple discussion, in the case of the plant cloning, it is done in horticulture, Fine. And see, in the case of plant cloning, which is done in horticulture, the best method of the plant cloning is all right. This is through vegetative reproduction. Fine. Let me elaborate here. This cloning in the case of the plant. Fine. Like see, this is the cloning in the case of the plant. This is plant cloning. This is plant cloning. This is plant cloning. Cloning in the case of the plant. So this plant cloning is done through cutting. And the cutting, what we are doing, we are just cutting the stem of this plant and then transferring to the new plant, and we are going to get the copy of the previous plant, right? So you heard about the cutting. Also, this is what it's a cutting. So you heard about so many ways to clone plant, like cutting, like grafting, like tuber, like rhizome. These all are asexual method of reproduction. And asexual method of reproduction is what? It's a cloning. One more example is also here. This is tissue culture. Right? This is tissue culture. So the ultimate purpose of what we are talking about, the ultimate purpose of the cloning in the case of the plant is to increase population in limited time. To increase population in limited time. Right? Like example, you heard about we are simply grafting. See, this statement is given by Asim Kumar Mehto, right? So he is talking about the rose plant. You heard about if you want to raise the number of rose plant, and we have a good variety of the rose. So what actually we are doing, we are simply cutting the stem off. We are peeling the stem of the rose plant and then tying with the the animal dung and the husk, and then wrapping with the polythene, and then pouring water at least for the couple of the days, maybe 1 week, 2 weeks, and then after after 15 days, after 20 days, we are just trying to observe where the peeled part start rooting or not. If yes, so we need to detach this rooted part from the main part, and then we need to transfer to the soil, right? So ultimately we have a good variety of the rose plant, and we are supposed to increase the number of rose varieties in a limited period of time, maybe 10, maybe 15, maybe 100, right, as per the requirement. So here ultimately we are going to raise this good variety of the rose plant, not only rose plant. This concept is applicable in every case. Means suppose if I have Alfonso mango, and if I want to increase the population of this Alfonso mango, in that case, if we are going to follow the same procedure, like the peeling the stem, then covering with the coating this peeled stem with the animal dung and with the soil, animal dung, and even the husk, and then tagging with the polythene, and then pouring water at least for the couple of days. So from the single plant we are going to raise maybe 100, maybe 1,000 the plantlets, and these 1,000 plantlets are supposed to be transferred to the soil, for the proper growth and development. So ultimately the purpose is what? Purpose of the cloning in the case of, like, see, the same also we are doing in the case of the potato. Potato is the best example of the tuber. So in the case of potato cultivation, we are just taking a one potato and then cutting into the pieces and then transferring to the soil. So every cut piece is going to give a new plant of the potato. That is the best example. So the point is that ultimately this asexual method of reproduction in the case of plant, that is also part of the cloning. Intention is what? This we are doing in horticulture and the agriculture, and the intention is to increase a number of good varieties in the limited time. Is the ultimate purpose of cloning in the case of the plant. So cloning does not make a sense of a dolly, that we are going to elaborate. That is the unique case of the cloning in the case of the animal cells, in the case of the animal. But even apart from this, cloning is quite simple, it's a quite common procedure in the case of the plant, and ultimately the purpose of the cloning in the case of the plant is nothing but the only purpose is just to increase the number of good varieties in a limited period of time. Right? Okay. Fine. Then coming to this artificial cloning in the case of animal cloning in animal, like natural. So one zygote is cleaved into two pieces, and the result is what? Identical twins. Result is the identical twins. So identical twins are cloned to each other, and this is the best example of natural cloning. But here we need to discuss about artificial procedure to clone, artificial method of cloning in animals. Artificial method of cloning in animals that we need to consider. Fine, okay. Right? So when we come to this artificial cloning of an organism, so here the first successful clone is what? It's a dolly. And dolly is cloned mammal. Dolly is cloned mammal. Right. So listen. So this the best example is the artificial cloning, and this is what we are talking about, the dolly. Yes, dolly. So see here, dolly was the first successful case of the cloning, human mammal cloning, and that was in 1996, 1997. Clear? And this was by Ian Wilmot. Ian Wilmot in Roslin Institute, Scotland. So the dolly was developed by Ian. Dolly was made in 1996-1997 by Ian Wilmot in Roslin Institute, Scotland. That was the first successful mammal cloning. But that was the first successful mammal cloning. right? And in the case of Dolly, the technology used was SCNT. This technique was used in Dolly. That is somatic cell nuclear transfer. Somatic cell nuclear transfer technology. Somatic cell nuclear transfer technology. This technique was used for the cloning of Dolly. And this question was asked so many times. Right? So we are going to elaborate what actually this somatic cell nuclear transfer technology is. I am going to elaborate. But before this, not only Dolly. When we succeeded Dolly, then after Dolly we cloned a number of animals. So we have a successful track history of a number of cloned animals. And let me show you. See here. This is a list, and I have shared this list in the form of PPTs to everyone in the last lecture. So these examples have already been shared to everyone through the PDF in the PDF format in the last lecture. And see here, this is monkey in 2000. Gaur, the first endangered species, was cloned in 2001. So the best example of the cloning. See, the best application of the cloning is what? Through the cloning, we can increase a number of endangered species, right? We can increase a number of endangered species. This is the benefit of cloning. Yes, mule, that is the Idausium, horse, Pamita, water buffalo, Samarupa was first cloned. The first cloned water buffalo, and it was in India. Where exactly? In the India's Karnal, National Dairy Research Institute. National Dairy Research Institute, Karnal. This was cloned and Samarupa. So in India, the first successful cloning was done in NDRI Karnal. Then camel in jars was the first cloned animal, and even we also succeeded the first Pashmina goat, Noori, in 2012, where exactly in Sher Kashmir University of Agriculture and Science and Technology successfully. So this was the first clone in world. This was the first successful achievement in the world, and that was the cloning of the Pashmina goat, Noori. So my point is that not only Dolly, apart from the Dolly, we have cloned a number of animals. But in the case of human organism, cloning is banned in the case of human. Means you cannot clone me. You can clone my stem cells. Right? That is permitted. And that permission is, the intention of that permission is for the medication that we have discussed in the last lecture as a therapeutic cloning through the stem cell technology. Right? So when we talk about cloning in the case of human, human cloning, yes. So a whole body cloning is not permitted. Whole body cloning is not permitted. Means you cannot clone me. But you can clone my stem cells, and the intention of the stem cell is for the medication, right? So the whole body cloning means organism cloning is not permitted in the case of the human, but adult stem cell cloning is permitted adult stem cell cloning is permitted, fine. And the intention is what? Intention is therapeutic cloning. And the purpose of means the therapeutic cloning is nothing but it's a medication. Cloning for the medication. And we have discussed about the cloning for the medication in the last lecture as a stem cell technology that we are doing for the bone marrow transplantation, that we are doing for the organogenesis. We are heading towards the technology where the growth and development of the organ could be possible in lab as per the demand that already we have discussed. Fine. Okay. So coming back to the previous discussion, and this is the case of the dolly. And this case of the dolly is nothing but it's a step. It's a SCNT. This is the case of the dolly, and this technique is Manav, though it is not permitted, but is whole human body cloning is possible? Yes, possible. 100% possible. 100% possible. Not easily, and in very technical way. It's not easy task. The way we are talking about, we are supposed to be. Even cloning is not easy in the case of other animals, like what we are going to discuss about the dolly. In the case of dolly, we cloned 277 embryos, and out of 277 embryos, the one embryo was succeeded in the form of the dolly, right? So the success ratio in the case of the dolly was even 1 out of 277. So that would be possible in the case of human also. So possible. In the case of human, it is possible, because see, once we cloned, once we cloned the camel, once we cloned the endangered species, once we cloned the Pashmina goat, once we cloned the sheep, means we have cloned a number of mammal animals. We have cloned a number of mammal animals. So once we have cloned a number of mammals, so why this human cloning could not be possible? So this is easily possible. It is easily possible, and even a few years back it was claimed by a doctor. I have succeeded, I have cloned, but even he was not able to justify. So that claim was futile, and even if he was supposed to justify, so that was the violation of the International Convention, because as per the International Convention, the whole body human cloning is not permitted. So permission is possible. Means cloning is possible. And even the human will also survive. Human will also survive, obviously. So let me elaborate. Fine. See, this is the somatic cell nuclear transfer technology. So in this case here, it's a sheep number 1. White-faced sheep, and from the sheep, udder cell was taken outside. Udder cell means somatic cell. Somatic cell. Udder cell means somatic cell. That is non-sexual cell. Right? Non-sexual cell. And this udder cell especially is what? It's a milk gland cell. Right? Milk gland cell. The cell we extracted from the breast of the sheep number 1, right? And that sheep is white-faced sheep. So this is what? It's a cell. And from the cell, we have just taken the nucleus outside. We have just taken nucleus outside. Means we have simply removed cytoplasm. Cytoplasm was removed. Cytoplasm of this cell was removed. Right? The second is hold your question. Hold your question for the 5 minutes. Hold your question for the 5 minutes. Let me complete, and then you start bombarding your question, and I am going to take one by one. At least let me finish first. Let me finish first, and then you please start asking your question. I am going to give you proper time to ask your question. So let me complete this entire process first of all, right? This is sheep number 2. Sheep number 2, black face sheep. So from the sheep number 2, what is extracted from the sheep number 2? Here, this egg cell is extracted outside. This is the egg cell of the sheep number 2, and you see here the nucleus of the egg cell is removed. The nucleus of the egg cell is removed. Means we have only cytoplasm of the egg cell is required. The egg cytoplasm, only the egg cytoplasm is required. So here we have one cell whose nucleus is required, and we have second cell whose cytoplasm is required. Right, which cytoplasm is required, and then see here, these two cells are fused with the help of electric shock. So see here, look here, this is the egg cytoplasm, and in this egg cytoplasm, this nucleus from the somatic cell is fitted like this, and as a result of this, we are going to have this fused cell, and in this fused cell, this nucleus is from adult cell, and this cytoplasm is from egg cell. Yes? Right? Then these fused cells begin to divide normally. Means we stimulated for a division. And once it started dividing, it means it behaves like embryo. Then we need to transfer to foster mother. Foster mother means surrogate mother. Surrogate mother. And this surrogate mother is sheep number 3. Sheep number 3. So as a result of completion, we are going to get this baby. We are going to get this baby, and this baby face is exactly similar to this first sheep. Exactly similar to first sheep. Clear? So the first sheep is nucleus donor. nucleus of somatic cell. This second sheep is cytoplasm donor, which is for nutrition. Yes, and this third sheep is the foster sheep, which is for embryonic development. So this baby is going to be born, and this baby will be exactly similar to sheep number 1, and this technique is what? It's a somatic cell nuclear transfer technique. Somatic cell nuclear transfer technique. Somatic cell nuclear transfer technique. Right? Fine. So again repeating. We need to remove nucleus from the somatic cell. We need to remove nucleus from the somatic cell, that is the non-sexual cell. Okay? And this nucleus of the somatic cell is inserted in the egg cytoplasm, means even the one egg is extracted from the other sheep, and the nucleus of this egg is removed, and then the nucleus of the somatic cell is inserted in the egg cytoplasm of the sheep number 2. Fine. And then we need to stimulate. Now this is what? It's an artificial zygote with the involvement of the nucleus from the somatic cell and the cytoplasm from the egg cell, and the intention of the egg cytoplasm is the nutrition. Then we need to stimulate, we need to induce this cell for division, and this induction is done by providing sufficient amount of nutritional material, by providing all conditions artificial which is required to stimulate cell division, right? As well as we need to induce expression of gene artificially, and once the cell starts dividing, it means this cell is acting like embryo. Fine. So now this dividing cell is acting like embryo, and then we need to transfer this embryo to the foster mother for the completion of this entire embryonic development. And once this embryonic development is done, the baby is going to be born like with the normal feature, with the similar feature as the sexually derived baby. And here this baby, the cloned lamb, is going to have gene from only one source, and that source is what? It's a nucleus. It's a nucleus of the cell. Is the logic behind clone is always monoparental in nature. Clone is always monoparental, because clone has only one. This cloned lamb is carrying one source of the gene, and that one source of the gene is what? It's a nucleus of the white faced sheep. So somatic cell nucleus is transferred, and as a result of this, the lamb developed, and that lamb is carrying the 100% gene similar to the nucleus of the white-faced sheep. And this technique is what? It's a somatic cell nuclear transfer technique. I'm just checking my internet is good. Just hold. All okay. From my side, there's no problem. If someone is not getting reception, 1 or 2 fellow, from my side everything is okay. From my side, everything is okay. Clear exception. Why there is a need of the third sheep as a surrogate mother? Listen, Siddhant and the Ritik. Now this we succeeded first of all. In that case, we involved three different sheeps. But now this technology is well developed this time. This technology that nowadays we are completely familiar about the technology, right? So in that case, suppose if you wish to clone me, which is not permitted. If you wish to clone me, which is not permitted, suppose if you are willing to clone me, which is not permitted this time, right? Human cloning is not permitted, but I'm just talking about the hypothetical aspect. So if you want to clone me, you take my any cell from anywhere, from any body part, except my gametic cell, and in my body part, the gametic cell is the sperm cell. Right? You take my hair cell, you take my skin cell, you take my heart cell, you take any cell from anywhere, right? And get the nucleus of, suppose if you have taken my hair cell, right? Just take my hair cell or just take my skin cell and take this skin cell is the non-sexual cell, because this skin cell is not involved in the process of the sexuality, because the sexual cells are involved in the process of the continuation of the generation, right? So the skin cell is not involved in the process of sexuality. Right? In that case, you take my skin cell is the non-sexual cell, somatic cell, and get the nucleus of this skin cell outside. Fine? Then we need to take the egg cytoplasm, because the egg cytoplasm is the richest source of nutrient. So you just take a female, any female, right, and get the egg of the female outside from the body of the female, right? So we have simply enucleated, or we have simply taken the egg of a female, and then we have simply removed the nucleus of the egg of that female, and now this time we are going to transfer the nucleus of my skin cell to the cytoplasm of the egg cytoplasm of the female, right? Fine. And now once this cell starts multiplying, so at the same time we are supposed to transfer that artificial cell to the uterus of that female for the completion of the embryonic development, right? So the clone, so after the 9 months of the gestation would be similar. The embryonic development would be similar, the embryonic development would be similar in the case of cloned baby of human as compared to the sexually derived baby of human, right? So the embryonic development period is similar. So after 9 months, you are going to get individual, and once this individual start developing, so that individual is going to resemble characteristics which is 100% similar to me, because that individual is carrying my 100% of the gene similarity, because the only source of genetic material in the case of that individual is what? It's my non-sexual cell, right? So in the nowadays, if you want to make my clone, for my clone there's an involvement of even at least one female, and that female would be acting like egg donor, and the similar female could also be act like foster mother, right? In the case of female clone, suppose if I want to develop a female clone, right, in that case what we are supposed to do, suppose there is a female, right, named as X female, we need to take the skin cell of the same female X, right, get it enucleate, means just take the nucleus of the skin cell outside of the female X. Again we need to take the egg of the same female and get remove the nucleus of the X, egg, and then you please transfer the nucleus of the skin cell into the cytoplasm of the egg cell, means we are going to develop an artificial egg with the involvement of the nucleus of the somatic cell and the cytoplasm of the egg cell of the same female, same female. And then you please transfer, after doing this, after inducing the division, you please transfer, right, in the same female, and that female is going to develop baby, right, develop baby. So in the first case, means once it was succeeded, right, we took 3 different sheeps, just for the caring and just to see how this character is moving. But now, after doing this entire exercise, we understood that this could be possible even if we are going to take only one female sheep, or even we are going to take one female and the one male, where the female would be, right, acting like the donor of the egg cytoplasm and the foster female, and the male would be acting like the somatic nucleus donor, all for the female itself. Right, no involvement of the male, rather the same female could be acting like a nuclear donor, the similar female could be acting like the cytoplasmic donor, and the similar female could be also acting like foster mother. So nowadays it become possible, right? But when we succeeded, it became, we involved 3 sheeps just for the confirmation whether we are going to be succeeded or not. And if we are going to succeed, we just wanted to see how this character is being moved from one sheep to other sheep. So that's why we involved 3 sheep. That was the experimental aspect. No, embryonic development can never be done in lab. This question already I replied even few lectures before. Embryonic development is always natural, whether it is the embryonic development of a human or even non-human animal, because the embryonic development is always supported by hormone, and that hormone is the influx of the female. Right, that female is going to secrete in the uterus is required for a development of. Fine, so artificial development is not possible. Development is always natural. Mamta, we have discussed, already we have discussed, we can also take the nucleus and the cytoplasm from the same sheep. The source of the nucleus would be the somatic cell. The source of the cytoplasm must be egg cell. Right, again you just try to construct it and then transfer to the uterus of the same sheep for the completion of the development. Yes, Mamta, there could be the chance of the appearance of the defect. There could be the chance of the appearance of the gene defect. This is the negative aspect of the cloning. In the case of cloning, right, the clone baby is going to have only one source of the gene. So in that case, there could be the chance of the expression of the defected gene. That is the biggest drawback of the clone. Varun is asking about why we are supposed to create an artificial egg by diffusing by injecting the nucleus from the somatic cell to the egg cytoplasm. Why we are not going to take the the cytoplasm and the nucleus of the same cell, like the egg cell. See, why? Because, well, we have discussed this concept earlier, but even I am going to tell you. Once again, I am going to repeat you. Because see, the sperm and the egg cells, sperm and the egg cells are the two cells in the body, and these two cells have only half of the number of chromosomes. Means my every cell has 46 number of chromosomes. My every cells have a 46 number of chromosomes. So if you take my all body part, I was derived in the form of zygote, and that zygote was carrying a 44 plus, means my genetic composition is 44 plus XY. That started multiplying. So throughout the body part, I have 44 plus XY number of chromosomes. But when it comes to my sperm cell or even the egg of the female, so the number of chromosomes is just half in the case of the egg and the sperm. Right? Egg and the sperm. So the nucleus of the egg has half of the number of chromosomes, that is 22 plus X. And the nucleus of the sperm has half of the number of chromosomes, that is either 22 plus X or 22 plus Y. So if we are going to use the nucleus of the sperm or the nucleus of the egg, fine. So the baby is going to have half of the number of chromosomes, and that would be 23 number of chromosomes, right? And I told you life is possible. For the consistency of the life, the number of chromosomes must be consistent. So the baby is going to have my all features. Once the baby is going to have my all chromosomes of genetic material, and the source of all chromosomes of the genetic material is what? It's my non-sexual cell. So why we are not using the nucleus of the sperm and the egg cell? Just because of the half of the number of chromosomes in the egg and the sperm cell, right? So we are discarding the nucleus of the egg cell. Why? Because this consists of half of the number of chromosomes. And why only cytoplasm of the egg cell? Because that egg cytoplasm is the richest source of the nutrient, and then this egg cytoplasm is being penetrated with the nucleus of the somatic cell. Why? Because the nucleus of the somatic cell has 46 chromosomes in 23 pairs. That's why only somatic cell nucleus is used for the cloning, not the sexual cell. I hope you understand well. Namrata, I hope you got an answer. Why not the sperm is used? Ritik, I hope you got your answer. Why not the sexual cell is used in the cloning? Anjana, some of the gene are also present in the cytoplasm, not in the cytoplasm. There is no gene in the cytoplasm, right? The insignificant number of gene sequences are reported in mitochondria. Fine. So now your point is genuine. So 99.9% of the genetic similarity could be possible between the cloned individuals and the nucleus. But 0.1% of the differences could be possible because of the mitochondrial gene, right? So as a whole, when we talk about this, we are simply avoiding this. That's why we are just talking about the clone is exactly similar to the parents. But this similarity could be 99.9% of the gene similarity. Ritik, you got your answer? Why we took two cells from the two shapes? Just because it was the first experiment. Siddhant, why we need to take a third sheep as a surrogate? I hope. Ayush, it will have all chromosomes from the sheep number 1. Yes, because the entire nucleus is being transferred. There is no fertilization, Gaurav. Mamta, I told you why the sperm cell cannot be used. Because the nucleus of the sperm has half of the number of chromosomes. The nucleus of the egg and the sperm cells have half of the number of chromosomes. That's why these two are not being used. And even when we talk about the sperm cytoplasm, the sperm cytoplasm has least amount of the nutritional material. So that is not going to support, right, this process of the division. That's why egg cytoplasm is only being used. Because see, as a whole, if you just look at in the normal process of the menstruation, this female, suppose if we talk about the human, right, human female menstruation. So one menstruation in human female, it results one functional egg. And the one functional egg has sufficient amount of nutrient, and even this one functional egg is fertilized. So that egg is going to be sustained at least for a couple of days, maybe 10 to 12 days, without getting any nutritional assistance from the mother. Right? Without getting assistance from the mother, this zygote, this fertilized egg could survive at least for the 10 to 12 days, because this egg has sufficient amount of nutrient. But one ejaculation of the male releases millions of the sperm, and the millions of the sperm is what? Even the amount of the nutritional material could also be distributed as per the number of cells. That's why the egg cytoplasm is used for the nutritional purpose, not the sperm cytoplasm. And the nucleus of sperm and the nucleus of the egg, right, can never be used just because of the half of the number of chromosomes. Namrata, I hope you understand. Yeah. If same female somatic and the egg cell is used, in that case even the mitochondria could also be same. No need to be sorry. Well, if you have any doubt, you are supposed to ask me, right? That is your right. If you have any doubt, and the doubt is genuine, I am going to answer. I hope you understand. No. The cloned sheep has similarity only with the sheep number 1. Only with the sheep number 1. Right? Only with the sheep number 1. Insignificant similarity with the sheep number 2, that even we can never justify in the normal course. And there is no similarity, there is no even the insignificant amount of the similarity we can never see between the foster mother and clone mother. So 99.99% of the similarity between the clone sheep and the sheep number 1, 0.1 or 2.01% of the similarity between the clone sheep and sheep number 2, just because of the mitochondrial gene, and 0% of the similarity between the foster mother and and clone baby, because the foster mother is going to provide space only for the embryonic development. Right? Yes? Asim is asking about. Asim, you asked this question already. We have discussed this question earlier. Can cloning help to treat cancer cells? Yes, that is the case of cloning of the stem cell. That is, this time we are talking about the cloning of. This time we are talking about the cloning of. It's a whole body cloning. What you are talking about, the cloning help to treat cancer cells? Yes, especially when we talk about the blood cancer. So in the case of the blood cancer, how this blood cancer is being treated? With the help of, by the bone marrow transplantation. So we need to take bone marrow from the donor cells, fine, and then we need to ensure the cloning of this stem cell, bone marrow. So ultimately the purpose of the cloning of this bone marrow stem cell is what? To increase the number of bone marrow. Fine. That is what? It's a therapeutic cloning. And then we need to transfer to the recipient end. So when we talk about that, already we have discussed in the last lecture. So that cloning which is done for the medication of the cancer cell is what? It's a therapeutic cloning or the stem cell technology. And this time we are talking about the third aspect of the cloning as a whole body cloning or organism cloning. Abhyar Asif Ali, can we use this technique in sheep and the goat, and what will be the... It's all hypothetical. It's all hypothetical. This way we are not supposed to talk about, suppose if I am going to clone human with the lion, and what would be possible. So that is that, we are supposed to answer this question once it is already been done. Otherwise, I am not supposed to make you the hypothetical answer. So it is not done so far what you are asking about. If it is done, so then I am supposed to answer. Namrata, see, listen, the meaning of the embryo is what? The meaning, listen, the embryo means the dividing cell, which is any cell that starts dividing, and that has ability to develop entire individual as an embryonic cell. Right? Any cell which has ability to develop, that start dividing and which has ability to develop the whole individual, that is considered as embryonic cell. Right? Now, usually embryonic cell results after the mixing of the sperm and the egg. That is the natural process. Usually embryonic cell results as a process of fertilization. That is the natural process of the embryonic cell. But even every cell has a similar genome sequences. Right? Every cell has same genome sequences. And suppose if I am going to take this skin cell, and in the skin cell only few gene sequences are active and the rest of the gene sequences are sleeping, in the sleeping inactive mode. And by any means, suppose if we are going to induce the gene expression of all gene sequences in the skin cell. So if the all gene sequences of the, suppose this gene, suppose this skin cell has gene sequences which is exactly similar to hair cells. Fine. So I told you that every cell has 20 to 25,000 of the gene sequences, right? So suppose if we talk about this human skin cell, this human skin cell has 20,000 to 25,000 gene sequences, and out of 20 to 25,000 gene sequences, only few gene sequences are expressible, active. It is possible for this skin. Rest of the gene sequences are inactive. Fine. And here in this process, what we are doing? We are simply inducing the expression of all gene sequences. Means we are just trying to activate. Activate inactive gene sequences. And when all gene sequences are going to be active, fine, then what happened? This skin cell will start multiplying, and this will be acting like the embryonic cell, right? And this skin cell, where the all gene sequences are going to be active artificially, it is going to be multiplied and it is going to develop all body part, because here the all gene sequences are active. So ultimately inducing cell to divide, that make a sense of the activating all gene sequences, and after activation of the all gene sequences, these gene sequences are going to be act like the whole body, and then the word embryo is being used for this particular kind of a cell, because here the all gene sequences are now become active, that we have induced artificially. But it is not possible the way we are talking about, right? Listen, in the case of the dolly, we made 277 cells like this, what we are talking about. Out of 277, we tried to stimulate the gene sequences of all cells, but out of 277, right, in only one cell, the gene expression was activated properly, and that was resulted in dolly. Clear? Otherwise, in natural case, what happened? The sperm is getting fused with the egg. The result is the zygote. The zygote starts dividing. That will become embryo, because this is the natural process. So naturally, we are not supposed to. There is no need to activate gene sequences, because it is naturally designed. So once the embryo is deriving naturally after the mixing of the sperm and the egg, so everything is natural, and this natural process is going to activate genetic sequences, and that genetic sequences are going to be expressed in developing embryos, and the embryo is going to develop an individual. That is the natural process. But artificially, if we are going to perform this entire discussion artificially, in that case we need to do effort, and that effort is what? That effort we were tried for last so many years, and then we succeeded in 1996 in the form of dolly. So this is hard. This is, in fact, it's a tough process. It's a technical process. It's a quite technical process, and the technical process, in the case of technical process, the chance of dejection is always more as compared to the chance of, right, acceptance. Fine. So that's why the success ratio of the cloning is not up to the mark, or as compared to the success ratio of embryonic development in the case of sexually derived individual. Yes, can we go ahead? Can we go ahead? Namata, I hope you understand. Yes, sweetie. See, obviously, every month female is followed by menstruation. Every month female is followed by menstruation, and every cycle of the male is followed by this spermatogenesis. So then what happened? There is a sperm mother cell in male, and there is an egg mother cell in female. Fine, and this sperm mother cell is followed by spermatogenesis process. So sperm is a product. Sperm is a product. Whether this sperm is going to be used or this sperm is going to be wasted. Fine, that formation process means the formation will continue. And how this formation is continue? This formation is continue, right, only once there is a stem cell for the sperm and the stem cell for the egg. But that stem cell is not going to be sustained throughout the life. There is a pause. After a certain stage of the life, these cells are going to be degenerate, and these cells are going to be degenerate, and then we are heading toward the sterility or the menopause in the case of the female. Asif, we are talking about the cloning. You are talking about the hybridization. There are two different perspectives. We are just talking about the cloning, and you are talking about the hybridization. You are talking about the tomato and the potato plant leads to tomato, right? There's also one more example like the Raphanobrassica, that is the reddish and the mustard leads to Aphanobrassica. See, this is what? It's a fertilization process. It's a fertilization process. Means we are simply mixing, we are simply showing the fertilization, means the cross breed between two different species. One species is the tomato and the second is the pomato. So we are simply inducing fertilization. It's the artificial reproduction. We are simply involving the two different plant gene sequences getting fused with each other, and the result is the pomato or even something else. Right? So here the result would be biparental. One source of the gene is the pomato and the one source of the gene is the potato. Fine, but the clone is always monoparental. Right? Why we talk about this pomato and the potato in the case of the plant? Again, you heard about the tigon and the liger. That is also possible in the case of animal. Right? Tigon means what? It's a male tiger and the female lion. The cross breed of the male tiger and the female lion. That is the tigon. Right? The cross breed of the male lion and the female tiger is the liger. And these two are the result of the interspecific hybridization. So, you are talking about the pomato in the case of plant, or even I am telling you lion and the tiger, these two are the result of the hybridization, and the hybridization is fertilization, the process of fertilization artificially, right? It's a process of the sexual reproduction. But what we are talking about, it's a asexual, right? And the asexual means what? The individual is going to have only one gene sequence. Yes, Riddhima, correct. So Asif, I hope you understand. Allow me to proceed. Right. So see here, if you see the advantages of the cloning, these are different advantages of the cloning. A possible answer to infertility. Yes, the treatment of the disease when we talk about therapeutic cloning. Yes, organ transplantation, cell or tissue on demand, that is stem cell, means cell based therapy. Yeah, designer baby means genetically upgraded baby. Yes, a better understanding of the genetic disease and stopping the aging process. Skin regeneration. Fine? Right? Yes? Can you go ahead? But even if you see here, the disadvantages of the cloning, junkyard of the human part, means we are going to develop human part, we are going to clone human part, even it is not required. So that will be act like a junkyard. And a clone as commodities, clone by black marketing. And India means across the world, and we are just look at the black marketing, it's quite common. So ultimately we are going to create an organ deficit condition and then start black marketing. Yes, defective outcome, fine, just because of the one source of the gene. The loss of the genetic variation because we are going to promote monovosity, which is against the against biodiversity. Sex determination could be done if you are going to, if we are going to clone myself. It means without doing anything, Anna, you have already determined the sex of the baby, because my genetic material will result individual which is similar to me. That make a sense of what? So that make a sense of the genetic determination of the baby. So sex determination is possible even without doing anything, and the false sense of authority, like suppose if you are going to clone someone, so we would be acting like we are acting like a god. Stopping the aging process means what? Ultimately, aging process is because of aging process is because of the loss of cells, right? Loss of cells. Fine. So see, suppose if you are going to induce the process of cell division naturally in your body, suppose if we have injected the skin cell in your body, and that skin cell is going to repair your degenerating skin cells. So ultimately your skin cells or the organ cells are going to be as intact. And that is what? That process is going to stop aging process. Aging process means what? The loss of cells are more as compared to the cell division. That leads to aging process. That we are trying to compensate. By cloning cell artificially and then transferring this clone cell to the body just to compensate the loss of the cell. Right? Fine. Can hair transplantation be a part of stopping the aging process? Here, there is no difference between the aging process and the hair transplantation. Right? Listen. Hair transplantation is something different. Hair transplantation is a stem cell transplantation. Aging is the loosening of the skin and the body part. The loosening of the skin and the body part that leads to aging process. Fine. So aging, in the process of the aging, we are just trying to compensate the loss of the cell in the skin and even the internal body parts. Aging is this is what it's aging. But the hair transplantation is even, so we could also see the baldness in the case of developing babies. Now it's a very common. So developing babies are grown baby. So even we have seen, we could also see the baldness in the case of the grown baby. So that is not aging process. That is just because of so many other conditions. हाँ भाई गो, प्लीज रिपीट द disadvantages point once again. यार गो ये बताओ, suppose if you are going to clone someone, if you are going to clone me, ठीक है, if you are going to clone me, so आपको ऐसा you will be acting like, you will start thinking like I have just created this man, ठीक है, I have just created this man, so I would be acting like God, है ना? I would be acting like God, and that is going to create the false sense of authority. Yes or no? Yes or no? Hello? You have cloned me. It means you have cloned male baby. You have cloned me. Right? You have cloned the 100 individuals like me. Means you have cloned 100 individuals like me. Means you have cloned 100 individuals like me, and all individuals are male. So this is going to create monomorphicity. And for the natural balance, there must be the female also. There must be the female. So if I am going to increase the male population, so definitely what happened? Somewhere we are lagging behind in the female population. Yes or no? ठीक है? क्यों भाई? बताएं. So can we go ahead? I think Today class is smooth. There's no problem. Network is fine from my side. I hope you are able to, you are getting this audio and the visual reception properly. Even signal is okay from my side. Fine. Chaliye. So in the same continuation, look here. Now, the next heading is DNA fingerprint. DNA fingerprint. And in human genome project, I told you more than 99% of the genetic similarity we could see. More than 99% of the base sequences are similar in human. Clear? Differences lie in the rest of the sequences. Means less than 1% of the sequences. Right, and these sequences are there is a good statement from the Nikhil Singh. Everyone is running as smooth as a knife through the butter, sir. Fine. Nowadays life is all about the butter and the corona is acting like a knife. Fine? Okay. So see, there's a less than 1% of the genetic differences. So this less than 1% of the genetic differences are nothing but it's a VNTR. And this VNTR we are trying to print. So it's not a printing of 99% of the bases. These 99% of the bases are not being printed because these are same in everyone. Cloning is legal or illegal? Cloning is legal. It's a perfectly legal. It's a perfectly legal except the human cloning. Except the human cloning. Except the human cloning. This we are doing. If you see, I told you the cloning is already been done. It is already been done for the plants. The intention is the good varieties. This cloning is already been done in the case of animal, right? This cloning is already been done in the case of synthetic meat, what we have discussed in the last lecture. That is also the concept of the cloning now. That is also the concept of the cloning. That cloning is the stem cell technology. The cloning is legal. But when this cloning comes in context to the human, right? So then we have so many criteria and the regulation. We have so many criteria and the regulation where the whole body cloning is illegal. We cannot supposed to be. Even the embryonic stem cell is illegal, and only the few examples of cloning is permitted in the case of human, and that is the stem cell therapy that already we have done in the last lecture. Okay, right. So here, now more than 99% of the base sequences are similar in all individuals, and the differences lie in 1% of the sequences, and these differences are less than 1% of the differences, right? That we need to print. And this is what it's the VNTR. Yes, fine, VNTR. So the point is that here, see, look here, the technique of the DNA fingerprinting was initially developed by Alec Jeffreys, and he used satellite DNA as a probe that shows very high degree of polymorphism. This is called as VNTR. VNTR means less than 1% of uncommon DNA. And why this is called as satellite? Why? Because this consists of repetitive bases like 80, 80, 80 or AA or GC, GC. So these sequences are repetitive. That make consist of the repetitive sequences or satellite DNA sequences. Satellite DNA sequences. Fine. So listen, look here. Now just try to understand what actually we are doing in this entire process. It's a complete technique of DNA fingerprinting. Let me elaborate. Here what we have, this is blood sample, means it's a cell, source of DNA. It's a source of DNA. Right? Now we need to take the nucleus from the outside. And this nucleus is the source of DNA. So the DNA of the nucleus is isolated from the blood sample. The DNA, this cell has nucleus, and the nucleus has what? Genetic material or the DNA. So this DNA we need to take outside like this. And then this DNA is subjected to restriction digestion. This restriction is what? This restriction digestion is, it's a restriction endonuclease enzyme which is acting like molecular seizures. Fine, and as a result of this, see, we have simply cut down. The intention is to cut VNTR. So, see here, listen, this is the first process. It's the first step, and this first step is the isolation of DNA. And this DNA is what? It's the isolation of VNTR. So, it's the isolation of DNA, then the digestion of DNA by restriction endonuclease. And this DNA is nothing but it's a VNTR. And this restriction endonuclease is molecular seizures. And then separation of the DNA fragment by gel electrophoresis, like gel electrophoresis. See, this is the process of gel electrophoresis. This is the process of gel electrophoresis. It's a gel. It's a gel. It's a agarose gel. This point is here, this agarose gel, this point is positive charge and this is negative charge. And the DNA starts moving like this because DNA is negatively charged. DNA is charged particle, negatively charged particle. DNA is always considered as negatively charged, right? So it's a plate of agarose. It's a plate of agarose where the DNA is stored like this, and then both the ends are connected through the electric wire, and then right current is being passed. So the negative charged DNA will try to move towards positive charge, and just because of the difference of the size of the DNA, we are going to get the separate VNTR on the agarose plate. Fine. Now see, this is what the transfer of DNA fragment to the membrane. See here, this now in this part, we could see the two different plates like this. One plate is like this, that is the below plate, and the above plate is like this. So the original plate, that is the result of gel electrophoresis, is like this, right? And here above this we need to put one more plate, and this plate is what? It's a nitrocellulose paper, and the intention of placing this nitrocellulose paper over the gel electrophoresis is what? Now this nitrocellulose paper will suck. It will suck DNA fragments from this agarose gel to the nitrocellulose paper, and this is a kind of blotting. Right? So this blotting technique was developed by Southern, E.M. Southern, and we named it as Southern blotting technique. So see here, the transfer of DNA fragment to the membrane, and this is what? Here, this membrane is nylon membrane, and as a result of this, see here, as a result of this transferring, now this DNA is going to have this DNA. We can see this DNA on nylon membrane, and this technique is called as Southern blotting. That we could see here. The transferring, it is mentioned as a transferring the blotting of separated DNA fragment to the synthetic membrane, such as nitrocellulose or nylon membrane, and this entire procedure is called as Southern blotting. This entire procedure is called as Southern blotting. Fine, right? Now, hybridization of these DNA segments, these are the VNTR. These are VNTR DNA. Hybridization of these VNTR with the radioactive probe. Radioactive probe means it's a artificial DNA with isotopes. Artificial, right? Artificial DNA with the isotopes. Fine. Artificial DNA with the isotope is radioactive probes, like this. Like this.